For the regulation of GI function, three aspects are there. Endocrine regulation, in which GI hormones are released into the circulation. Paracrine regulation, in which the hormones or these chemicals are released from nearby cells and they act on the cells which are just nearby. For example, the release of histamine, which acts on the parietal cells. This we have already seen in the video on gastric acid regulation. Then there is neural, which we have extrinsic and intrinsic influences. Now, these three modes, endocrine, paracrine, and neural, they basically regulate the functions of the GI tract, that is the secretion, digestion, absorption, and motility. So, they are released. Basically, the main stimulus is when the food enters a particular region of the GI tract, and whatever is the function of that particular region of the tract, that is brought about by the release of these hormones. So we will see, if you understand this fundamental concept, we are able to remember the function of the GI hormones better. Now, these GI hormones are the polypeptides and are important for the regulation of gastrointestinal secretion and motility. They are secreted by the cells known as endocrine cells. Entero means present in the GI tract, and these are the endocrine cells. So, endocrine cells. There are many names given to these cells, such as I cells, K cells. We will see as we progress with each hormone. And these endocrine cells are present in the mucosa of stomach, small intestine, and colon. Many of these secrete only one hormone. However, some synthesize serotonin and histamine, that is, these are the amines which are synthesized, and these cells where these amines are synthesized, they are known as enterochromaffin cells. Okay, so ECL cells release serotonin and histamine. This we have already seen again in the video on gastric secretion. Now, these GI hormones, based on the structure and function, there is some similarity in structure and function. So they are classified mainly into two families, and these are the Gastrin family and Secretin family. And there are others which cannot be classified in any of these families, so they are kept into others. So in Gastrin family, we have Gastrin and CCK, which have similarity in structure. Then in secretin family, we have 5 hormones: secretin, glucagon, glycine, VIP, and GIP. And rest of the hormones all fall in others category. Now, what is the site of release of GI hormone? This diagram is taken from Ganong, and it is showing the major site of the release of the hormone and minor site as well. So if you see gastrin majorly is released from antrum. The breadth of this arrow, it is showing the major site of the release, and then it is tapering. So it is also released from duodenum and jejunum, but the amount released is decreasing. From the antrum, maximum amount is released. Then CCK and secretin, again duodenum and jejunum. GIP and motilin only from duodenum and jejunum, but we will see later that motilin is also released actually from stomach, that Ganong only has written, but it has missed from this diagram. Okay, so we will discuss only about the major site of the release. So when we talk about the gastrin, we will talk that it is released from the antrum, right? Similarly, CCK and secretin, we will talk that it is released from the upper small intestine. Now, moving on to each of the hormone. First hormone is gastrin. Gastrin is released by enteroendocrine cells known as G cells. G cells which are present in the antrum, and this gastrin release has various size depending on the number of amino acids which are present. I told you that this GI hormones are polypeptides. So they are like this, lot of amino acids are there, and number of the amino acid decides the size of the polypeptide. So gastrin is released in various sizes. We have G34, G17, G14. For the function of the release of gastric acid, it is the G17 which is most important. So here we are seeing one G cell, and you see that the apex of this G cell has receptors. And local amino acids and peptides, that is, which are present in the stomach, bind to these receptors and cause the release of the gastrin. And that is true for all the enteroendocrine cells we are going to talk. So, on the apex, there are receptors to which certain chemicals can bind and cause the release of that particular GI hormone. So, what is the stimulus for release of gastrin? We have certain chemicals which are present in the food and they are present locally in the stomach. Then we have mechanical stimulus, that is the distension of the stomach. Then we have the neural stimulus as well, that is the parasympathetic nerve supply, where vagus releases gastrin releasing peptide and not acetylcholine. The gastrin releasing peptide acts on the GRP receptor and causes the release of the gastrin. Then from blood, we have some stimuli, that is the calcium and epinephrine. So you see the locally which stimuli are acting, that is if they are present in the stomach, they are acting on the apical side, and those coming from neural and from the blood, they are acting on the basolateral side. Actually, this distension of the stomach, it will cause the stretching and ultimately it will cause the stimulation of the neural supply as well. But directly also there is some action and causes release of the gastrin. Now this gastrin enters into the blood. Via blood, it reaches the parietal cells. So parietal cells are present in the fundus only. But gastrin is released into the blood and it reaches to the parietal cells via the blood. It acts on the CCKB receptors. It is not A, it is B receptor, which is present on the parietal cells. So I told you before that gastrin and CCK are of the same family. So that is why there is some receptor homology as well. So this gastrin acts on CCKB receptor and causes release of hydrogen ions. This hydrogen ion now has a negative feedback effect on gastrin release. So hydrogen ion either directly inhibits the G cell or it acts on D cells and causes release of somatostatin, which also causes inhibition of the G cells. Then there are other stimuli also which inhibit the secretion from the G cells, that is secretin, GIP, VIP and glucagon. We have seen in the video on gastric secretion that secretin is having a inhibitory effect on hydrochloric acid release. And why is it? Because secretin inhibits the G cells which cause decreased release of the gastrin and hence decrease in release of HCl from the parietal cells. Okay, so these are the inhibitory effects. That is a negative feedback effect mainly from the hydrochloric acid which is released from the parietal cells. So negative feedback, it inhibits the gastrin, and also there are hormones which are released in the small intestine. They enter into the blood and they also inhibit the gastrin secretion. Now, in pernicious anemia, there is elevated gastrin levels. And why is it happening? Because in pernicious anemia, there is loss of this parietal cells. Atrophy of gastric mucosa occurs and parietal cells decrease in number. So when parietal cells decrease, there is decrease in the release of the hydrochloric acid. So hydrogen ions are not able to inhibit the G cells. Hence, there is increase in the release of the gastrin. So in pernicious anemia, we have chronically elevated gastrin levels. So this is the table which is given in Ganong, enumerating the stimuli that increase gastrin secretion and stimuli that inhibit gastrin secretion. If you see, we have dealt all of them, right? Moving on to what are the functions of gastrin. For remembering the functions of GI hormones, if you remember a simple dictum, you will be able to remember the function. So when we talk about gastrin, main function is digest food in stomach and move it to intestine. So based on this dictum, if you try to remember what will be its function, you will see that it should increase the stomach secretion. So yes, it increases acid and pepsin secretion. It increases gastric motility because the food should digest and move into the small intestine. So it increases acid and pepsin secretion. It increases gastric motility. It also causes a stimulation of growth of mucosa of stomach, small intestine and large intestine. So it has a trophic action as well. Trophic action. Okay. Now moving on to the next hormone, that is cholecystokinin. Cholecystokinin is released by eye cells which are present in the mucosa of upper small intestine. Initially, actually, it was considered as two hormones CCK and PZ, that is cholecystokinin and pancreasamine. Now it is realized actually it is only one hormone, that is cholecystokinin. So some places you will see it as written as CCKPZ. It is the same thing. Okay. So what is the fundamental function of cholecystokinin? Digest food in intestine, move it further and prevent overloading of small intestine from stomach. Okay, because food or the chyme is entering from stomach into the small intestine, we want to digest it, and till the time that digestion is taking place, we don't want the small intestine to be overloaded with all the contents from the stomach. So based on this, if we see what are the functions of cholecystokinin, we will see that it causes decrease in the stomach secretion and decrease in the stomach motility, because there should not be any overloading of the small intestine with stomach contents. Then it causes pyloric sphincter contraction again, so that the contents from the stomach should not enter into the small intestine. Then for digestion in the small intestine, it causes increase in the secretion of the pancreas and the liver. So there is increased pancreatic enzyme secretion. There is contraction of the gallbladder so that more bile can enter into the small intestine, and relaxation of the sphincter of Oddi. Sphincter of Oddi we have seen in the pancreatic secretion that it is present at the ampulla of the Vater. So here ampulla of Vater opens into the duodenum at the duodenal papilla. So somewhere here the sphincter of Oddi is present. So when this relaxes, then only the contents of the pancreatic secretion and from the gallbladder are going to enter into the duodenum. Okay, so relaxation of the sphincter of Oddi does. Then intestinal motility, it increases because food should digest and move forward. It also causes increase in the synthesis of enterokinase. Enterokinase is the enzyme which is released in a small intestine, which causes activation of trypsinogen to trypsin. So we want that after release of the pancreatic enzymes, they should be activated as well. So increase synthesis of enterokinase. Then it has trophic actions on pancreas, and it also potentiates the action of secretin. This we have already seen in the video on pancreatic secretion, that how cholecystokinin and secretin potentiate each other action, and that is also because of their mechanism of action. They have different mechanism of action. Cholecystokinin acts by increasing the activity of phospholipase C, that mechanism, and secretin acts by increasing the CAMP. Okay, so both potentiate each other's action. Now, what is the stimulus for release of CCK? CCK is released from eye cells in the upper small intestine, and amino acids and peptides and fatty acids with more than 10 carbon atoms, which have entered into the small intestine from the stomach, that causes release of CCK from eye cells. CCK enters into the blood, goes and acts on the pancreatic acinar cells. So there is the receptor, CCK receptor. It acts on the pancreatic acinar cells, causes release of pancreatic juice rich in enzyme. Okay, rich in enzyme. And there is increase in the bile and pancreatic juice. Now, because the secretion is rich in enzymes, it causes further digestion of the nutrients, and there is further release of these products of digestion, which exert a positive feedback on the release of cholecystokinin. So, more and more products of digestion as they accumulate, they cause more and more release of CCK. In Gastrin, we saw that it was a negative feedback control. Here, it is a positive feedback control. So, more release of CCK occurs. Only when the products of digestion are absorbed by the intestinal mucosa and rest of the contents move forward, then the CCK secretion stops. Moving on to the next hormone, that is secretin. Secretin was the first hormone to be discovered by Bayliss and Starling in 1902. And after secretin discovery, they are the ones who called certain chemicals as hormones, which are released into the blood and act in a coordinated manner. This secretin is released by S cells in upper small intestine. And what are the function? Fundamental function is protect small intestine from excessive acid coming from stomach. That is the fundamental function. So if we see what will be its action, stomach secretion decrease, stomach motility decrease, and pyloric sphincter contraction, same as that of cholecystokinin. Because if it has to protect the small intestine from the acid, then it should prevent its overloading as well. So all this happens. Secretion, basically it promotes the increased secretion of bicarbonate from the pancreatic ducts and the bile duct. So increased pancreatic secretion which is rich in bicarbonate. So we say watery alkaline pancreatic juice is released when secretin stimulates the secretion. And secretin also potentiates the action of cholecystokinin. Okay, so I hope you will be able to simply remember that what is the functions of various hormones if you understand the fundamental functions of these hormones. Then, what is the stimulus for release of the secretin? Secretin, I told you, it is released from S cells in upper small intestine, and there is amino acids and peptides and acidic chyme which enters in small intestine. So, acidic chyme, this is very important. Acidic chyme, that is the presence of acid in duodenum, stimulates the release of secretin from small intestine, which enters into the blood and acts on the secretin receptor mainly on the pancreatic ducts, and it causes increase in the formation of CMP, promoting the release of bicarbonate. So alkaline pancreatic juice is released, and this has a negative feedback effect. This green arrow is wrong, it should be red. So it has a negative feedback effect because as the bicarbonate secretion increases, the acid in the duodenum is neutralized. So that is further going to decrease the release of secretin because acid is the stimulus. If it is neutralized, as secretin stimulus is going to decrease. Moving on to the next hormone, that is GIP. Remember that three fundamental hormones we have talked, gastrin, secretin, and cholecystokinin. They are the most important for remembering, but we have many other GI hormones as well. So we have glucose dependent insulinotropic peptide. Insulinotropic. Other name for this before it was gastric inhibitory peptide GIP. But later on we found that it has some different function as well. So this GIP is released from K cells in jejunum and duodenum, and stimulus for its release is glucose and fat in duodenum. So, with the presence of glucose and fat in duodenum, this GIP is released and enters into the blood and causes the release of insulin from the beta cells of the pancreas. So, even before the glucose is being absorbed into the blood, this GIP is causing release of insulin. So, it is more like an anticipatory increase in the insulin secretion for the preparation of the body to act against glucose. Okay, glucose has not yet been absorbed, so before itself insulin levels start rising, and this is known as incretin effect. Incretin effect. Okay, so we can say anticipatory increase in insulin secretion is brought about by this GIP. And what is the other function? It inhibits gastric motility, as was its name before, gastric inhibitory peptide. It also inhibits gastric secretion, and very important, it stimulates insulin secretion. So this is a organized diagram taken from Ganong where it says that food in the stomach causes gastrin secretion, and it causes increased acid secretion and increased motility. So you see how it is linked with the food in the stomach and the function of the structure. So food in the stomach is causing gastrin secretion, which is causing increased acid and pepsin secretion and increased motility. Then the chyme enters into the duodenum, where it causes release of CCK and secretin, causing pancreatic and biliary secretion, and which causes intestinal digestion of food. Then it also causes release of GIP, which promotes insulin secretion. And there is release of another hormone, peptide YY, which inhibits the stomach function, inhibits motility, inhibits acid secretion. But we have seen that CCK also and secretin also inhibit these functions of the stomach, right? Now moving on to some other hormones. Very briefly, we are going to see their function. Vasoactive intestinal peptide. It is released by nerves in GIT. And what is its function? See, its name is vasoactive. So it leads to vasodilation. For vasodilation, it causes relaxation of the smooth muscle. Yes, it causes relaxation of the smooth muscles of the blood vessels. It also causes relaxation of intestinal smooth muscle, and it also causes relaxation of the sphincters as well. Then, because it is causing vasodilation, it stimulates intestinal secretion of electrolytes and water. So, by remembering this vasoactive word, you can remember its functions. Then peptide YY. The stimulus for its release is fat in duodenum, and peptide YY inhibits gastric acid secretion and motility. In fact, they consider peptide YY as new gastric inhibitory peptide. So that name GIP, what they initially thought that the function is gastric inhibitory peptide, now they are moving it to peptide YY. The next hormone is ghrelin. You break down this word as GHRELN. You will remember its function based on that. Ghrelin, so it is GH. It is growth hormone releasing. Okay, so it stimulates the release of growth hormone. IN for intake. It stimulates food intake and it is secreted by stomach. So ghrelin stimulates the food intake, stimulates the release of the growth hormone. Then somatostatin. Somatostatin is released by D cells in stomach, and we have seen that how somatostatin decreases the gastric acid secretion. Somatostatin actually has inhibitory effects on all motility and secretion. So it inhibits secretion of gastrin, secretin, GIP, VIP, motilin. It is not motion, it is motilin. Inhibits gastric motility, inhibits gastric and pancreatic secretion, inhibits gallbladder contraction. So that is somatostatin. Statin is stop. Okay, stat means stop. So it is stopping so many functions. Then we have motilin. Motilin is released by MO cells in stomach, small intestine, and colon. So this I told you before in the diagram from the Ganong. They have shown that it is released only from the small intestine. But here we are seeing that it is released from stomach, small intestine, and colon. Insulin increases the GI motility in between meals. In between meals. Important. Okay. And that's why it causes migrating motor complex. This we will see in the video on GI motility. Okay, so it increases the GI motility in between meals, causing migrating motor complex, and hence it clears GI of undigested material. Now here, this table from Guyton is showing a summary of all hormones which we have dealt. If you see, you can pause the video. It is same as that we have already dealt. Okay. These are certain other small hormones. Again, we are not going to deal with it because there are so many chemicals which are being detected. We are not going to deal with it. Only one thing I will talk here, that substance P. Substance P is important for the perception of pain sensation. Very important. Okay, so that was all about the GI hormones. I hope that by remembering the fundamental function of GI hormone, you will be able to remember its actions.